Welcome to my channel, Journey to Education. Subscribe and follow reminder. Today, we're unlocking the next chapter in the secrets of our very DNA, diving deep into the principles of inheritance and variation.
So grab a seat and get ready for a truly fascinating deep dive. We're going to explore the biological blueprints that make us, well, us.
Okay, let's dive into this. Here is our roadmap for today. 1. Mendel's assortment and legacy. 2. Chromosomal theory of inheritance. 3. Morgan and genetic linkage. 4. Mechanisms of sex determination. 5. Mutations in the genetic code. And finally, 6. Mendelian and chromosomal disorders.
Let's kick things off with Section 1, Mendel's assortment and legacy.
You've probably heard of Gregor Mendel, right? Well, he formulated this absolutely foundational abstract rule called the law of independent assortment. Basically, it states that when you combine two pairs of traits in a hybrid, the way one pair segregates or separates is completely independent of the other pair. So, think about it like this. If you're looking at a plant's seed shape and its seed color, the way those shape genes separate has literally zero impact on how the color genes separate. It's important to note, though, that this law usually only holds up for genes that are located on entirely different chromosomes.
And this brilliantly illustrates that exact concept. Just take a look at the results of a classic dihybrid cross. This is where parents differ in two trait pairs, like, say, crossing round yellow seeds with wrinkled green seeds. When you actually map this out on a 16-square Punnett grid for the F2 generation, that independent sorting we just talked about perfectly yields this iconic phenotypic ratio of 9 to 3 to 3 to 1. You get 9 round yellow, 3 round green, 3 wrinkled yellow, and 1 wrinkled green. It's just beautiful, totally predictable math. But here's the really tragic part of the story. Mendel's absolute genius was basically ignored until the year 1900. Why? Well, first off, communication back in the 19th century was incredibly slow. The idea spread about as fast as a snail. Second, applying hard statistics and math to biology, that was considered completely radical back then. Plus, Mendel's whole idea of these stable, discrete genetic factors didn't really mesh with the continuous natural variation people saw every day in nature. And perhaps the biggest hurdle, he couldn't just point to something under a microscope and say, hey, look, there's the factor. It wasn't until scientists like De Vries, Correns, and von Tschermak independently rediscovered his work in 1900 that Mendel finally got the credit he deserved.
Moving right along to Section 2, Chromosomal theory of inheritance.
So fast forward to 1902. Scientists Walter Sutton and Theodore Boveri start noticing this incredible parallelism between Mendel's abstract factors and the actual physical chromosomes inside a cell. They realize that both genes and chromosomes occur in pairs in diploid cells, and they both segregate during gamete formation, meaning a gamete gets only one from each pair. Plus, independent pairs of chromosomes segregate independently of each other. It was exactly like Mendel's independent assortment.
Now, what's really interesting about this is how Sutton and Boveri connected the dots. They took the knowledge of chromosomal segregation during meiosis and literally united it with Mendelian principles. Their big light bulb moment was realizing that these genes physically sit on these segregating chromosomes. The two alleles of a gene reside at homologous loci on homologous chromosomes. And boom, the chromosomal theory of inheritance was born.
Section 3, Morgan and genetic linkage. Now, to actually prove this chromosomal theory, Thomas Hunt Morgan needed the perfect test subject. And he didn't go with pea plants. No, he used the tiny fruit fly, Drosophila melanogaster. And he chose them for some very specific, highly practical reasons. First, they grow super easily on a cheap, simple synthetic medium. They also have an incredibly short 2-week life cycle, so you can study multiple generations in a single month. On top of that, a single mating produces hundreds of offspring. The sexes are super easy to tell apart, males are noticeably smaller, and crucially, they have tons of visible hereditary variations you can see right under a basic low-power microscope. So Morgan starts crossing these flies, and suddenly he notices something really weird. When he crossed dihybrid flies, the resulting ratios completely deviated from Mendel's expected 9-3-3-1. What was happening? Well, he discovered that some genes were actually located on the same chromosome, meaning they couldn't assort independently. To explain this, he coined two vital terms. Linkage is the physical association of genes on the same chromosome, which basically causes parental traits to be inherited together. And recombination is the generation of new non-parental gene combinations, which happens because of crossing over. Morgan realized that tightly linked genes, like white eyes and yellow bodies in his flies, showed incredibly low recombination, just 1.3%. But if genes were sitting far apart, they were loosely linked and showed much higher recombination.
Let's move to Section 4, Mechanisms of Sex Determination.
Okay, so sex determination isn't just a one-size-fits-all rule across the animal kingdom. It largely depends on which sex produces two different types of gametes, a concept we call heterogamety. In humans and our fruit fly friends, we use the XY system. Males are heterogametic, meaning they produce both X and Y sperm, while females are homogametic, producing only X eggs. So in our case, the sperm determines the sex. But wait, it gets weirder. Take grasshoppers. They use an XO system where males are literally just missing a second sex chromosome altogether. And then it flips completely in birds with the ZW system. In birds, the males are homogametic, ZZ, and the females are heterogametic, ZW, meaning for birds, it's the female's egg that actually determines the sex of the offspring. But if you want a truly mind-bending system, we have to talk about the honeybee. They don't even bother with specific sex chromosomes. Instead, they use a haplodiploid system. Females, so the queens and the workers, are diploid. They have 32 chromosomes from a totally normal fertilized egg. But the males, the drones, they're haploid. They only have 16 chromosomes because they develop from completely unfertilized eggs through a crazy process called parthenogenesis. This literally means a male honeybee has absolutely no father, and he cannot have sons. But he does have a grandfather, and he can have grandsons. Honestly, nature's diversity is just astounding.
Next up, Section 5, Mutations in the Genetic Code. So the crucial point here is that our DNA is actually pretty fragile, and when it gets altered, we call those alterations mutations. At the gene level, there are two major types you need to know. First is a point mutation. This is a change in just a single base pair. For example, in sickle cell anemia, the DNA sequence GAG accidentally swaps to GUG. Just that one tiny letter swap changes the resulting amino acid from glutamic acid to valine, which completely alters the physical shape of red blood cells. Crazy, right? Then you have frameshift mutations. This happens when base pairs are either deleted or inserted. Because DNA is read in sets of 3 called triplets, inserting or deleting even a single base shifts the entire reading frame down the line. It totally scrambles every single piece of genetic information that comes after it. And finally, Section 6. Mendelian and Chromosomal Disorders. Genetic disorders generally fall into two broad classes. The first are Mendelian disorders, and these trace directly back to those single gene mutations we just talked about. To track how these pass down through generations, geneticists use pedigree analysis, basically a visual chart mapping traits across a family tree. By using this, we can trace disorders like sickle cell anemia. There's also colorblindness, which is an X-linked recessive trait, meaning it mostly affects males. Hemophilia is another X-linked disorder that causes blood clotting failure. You might know it famously spread through the royal families of Europe. We also have phenylketonuria, which is a metabolic issue where you lack a specific enzyme, and thalassemia, a recessive blood disorder. What all these have in common is that they trace back to one specific, localized error in the genetic sequence.
But then there's a second class, chromosomal disorders. Now, these aren't just tiny single base pair typos. These are massive, macro-scale failures of chromosome separation during cell division. For instance, aneuploidy happens when chromatids just fail to segregate properly, resulting in the gain or loss of an entire chromosome. This is exactly what causes conditions like Down syndrome, Klinefelter's, or Turner syndrome. On the other hand, you have polyploidy, which is an increase in the entire set of chromosomes. This one is actually a lot more common in plants than in animals.
Which leaves us with this final thought for today. When a single microscopic base pair change out of literally billions can completely alter the trajectory of a human life, how well do we truly understand the brilliance and the sheer fragility of our own genetic code? Drop your doubts and suggestions in the comments below.